Hello students, myself Shinali Shah welcomes you on behalf of Saraswat International Academy. Students, today we will begin with the next part of the chapter Principles of Inheritance and Variation. It is our part 11. Now students, in the last lecture we have studied regarding the process of recombination and linkage, right? In linkage we have also studied the two different types of crosses occurring in Drosophila. In one cross, we have studied that those genes which are located nearer to each other, basically they will show less percentage of recombination, and genes which are located distantly on the chromosomes, they show more percentage of the recombination, right? Now students, today we will begin with the next topic, and that topic is polygenic inheritance. Now students, what is polygenic inheritance? Now students, till now we know that a pair or basically a gene which occurs in a pair, a single gene pair, it is responsible for giving a particular trait, a particular character. Till now we have studied that. But students, there are certain character where not a single gene but more than one pair of gene, they combine together and give the effect. So what is polygenic inheritance? So several genes they combine to affect a single trait. That is, there are more than one pair of genes and they will give a trait or a character to an organism. So what is polygenic inheritance? A group of genes that together determine a characteristic of an organism is called as polygenic inheritance. So basically the group of genes will give not only a one pair of gene but more than one pair of gene together will give a character to an organism. What is that called as polygenic inheritance? Now students, here the intensity of character, it depends upon the number of the dominant allele. So what type of character and the intensity, it will be decided by the number of the dominant allele. This particular thing will be clear when we take the example of the human skin color. But before that, one another example. Now students, we have studied a pea plant, right? In a garden pea plant, we know that Mendel studied the height as a character. So in that the height case, there were only two possibilities. Either the plants were tall or the plants were dwarf. But now assume height as a character in human beings. Now is it like that either the humans are tall or dwarf, or do we see many height, right? So here we can say that in height, when we talk about the example in human beings, then there is a wide range of the possible height. So here generally such particular traits, they are controlled by three or more genes. And what are they called as? They are called as the polygenic traits. For example, again we'll read it. In humans, we don't just have the tall or short height people as the two distinct alternatives, but we have a whole range of possible height, right? Like pea plant, in pea plant there were only two possibilities, either the plants were tall or the dwarf plants. But is it the case in the human beings? No. In human beings there is a wide range of possible height. So basically such traits are controlled by three or more genes. And what are such traits called as? They are called as the polygenic trait. So here height is determined by more height or other such characters. They are determined by more than three genes. What are such traits or character called as? Polygenic traits. Now apart from the genes playing the important role, the environment also plays the important role in giving such traits. So it also takes into account the influence of environment. That is, not only the genes they give the character in such cases, but along with the genes, the environment also plays the role to give that character. Now student, we take the example of the human skin color to explain the polygenic inheritance. That is poly, it means many, multiple. Genic, that is genes. Many genes will control a particular trait. What is that called as? Polygenic inheritance. And a very good example of the polygenic inheritance is human skin color. Now students, human skin color is controlled by 3 different genes, and these 3 different genes are located on different chromosomes. And as these 3 genes are located on the different chromosomes, they are inherited independently, right? We know that those genes which are located on the different chromosomes, they assort independently, right? According to the law of independent assortment. So, the 3 genes are gene A, gene B and gene C, which are located on the different chromosomes. Now, assume that the 3 dominant capital letters, that is capital A, capital B and capital C, they control the dark pigmentation, because in this case more melanin is produced. So 3 genes, right? There are 3 pairs of genes. So out of that, the dominant genes, capital A, capital B and capital C, what do they control? They control the dark skin color. Why? Because they produce more amount of melanin. So basically, if dominant genes are there, it indicates the production of the melanin. And the recessive alleles of these genes, that is small a, small b and small c, they control light pigmentation, because lower, or you can say no amount of the melanin will be produced by them. Now students, these genes, capital A, capital B, small b, capital C, small c, they are located on the different chromosomes and are inherited independently. These genes, they are located on the different chromosomes and they are inherited independently. Now students, an individual which have a genotype with all the dominant genes, right? Listen very carefully. A individual with a genotype with all the dominant genes. So basically, what will be the genotype of that individual? Capital A, capital A, capital B, capital B, capital C, capital C. Now, in such individual, there will be the maximum amount of melanin and its skin will be very dark skin, right? Similarly, an individual with a genotype with all the recessive genes, that is small a, small b, small c. Now in them there will be lowest amount of melanin and their skin color will be very light. A individual with all the dominant gene will have maximum melanin production and their skin color will be very dark, and a individual with all the recessive genes have the least amount of the melanin and they will have the very light skin. Now student, listen very carefully. Each dominant capital gene produces 1 unit of color. So depending upon the intermediate skin colors are produced depending on the number of the dominant capital genes in the genotype. Now students, each dominant gene indicates the amount of the melanin produced. Listen very carefully. Each dominant gene will produce a particular amount of the melanin. So basically, we have the white intermediate skin colors. An individual may have 6 dominant genes, an individual may have 5 dominant gene, an individual may have 4 dominant gene, an individual may have 3 dominant gene, an individual may have 2 dominant gene, it may have 1 dominant gene or it may have no dominant gene. So, each dominant gene indicates the amount or unit of melanin produced. So, we have basically intermediate skin colors produced. Why? Because it depends upon the number of the dominant gene present. For example, a person whose genotype, so example, a genotype with 3 dominant alleles or 3 dominant genes and 3 recessive genes, that is in this condition, that is capital A, small a, capital B, small b, capital C, small c. So here there is a medium amount of melanin produced because there are 3 dominant genes and 3 recessive genes. So here medium amount of the melanin is produced, and this their skin color will be the intermediate skin color, that is their skin color will be neither extremely dark nor their skin color will be the extremely light skin color. They will have the intermediate skin color. This person with the intermediate skin color is called as a mulatto. So the latter genotype, that is the genotype where a person have 3 dominant gene and 3 recessive genes, that is a person with genotype capital A, small a, capital B, small b, capital C, small c, it will be considered as a mulatto, which means a person with the intermediate skin color. I told you earlier, each dominant gene directs the synthesis of same amount of the melanin pigment which gives the color to the skin. It means if suppose capital A gene is there, capital B gene is there or capital C gene is there, the amount of the melanin produced by all these 3 dominant gene will be same. Hence, we can say that the shade of the skin is directly proportional to the number of genes, dominant genes present in the individual. From this, we can conclude that the skin color is directly proportional to what? The number of the dominant gene. More the number of dominant gene, the darker will be the shade of the skin. So the shade of the skin is directly proportional to the number of the dominant alleles present in the individual. Now students, here there is a cross. See the cross very properly. So here who is taken as a parent? So pure white, or you can say a very light individual where there are only all the recessive genes, that is all the 6 genes are with genes, recessive genes, and that person is crossed with a very, or you can say extremely dark individual. So here what will be the genotype? Capital A, capital A, capital B, capital B, capital C, capital C. Now students, all the gametes produced by the very light individual will be small a, small b, small c, and all the gametes produced by the very dark individual will be capital A, capital B, capital C. So basically all the individuals of the F1 generation will have the genotype capital A, small a, capital B, small b, capital C, small c, which is considered as an intermediate condition, and that is a characteristic of mulatto. What is it called as? Mulatto. Now students, when there is an intercrossing between the F1 individuals, so basically here it is a trihybrid cross, so number of gametes produced will be 8. So basically these are the 8 types of the eggs produced, and these are the 8 types of the sperms produced, right? Capital A, capital B, capital C, small a, capital B, capital C, capital A, small b, capital C, capital A, capital B, small c, small a, small b, capital C, capital A, small b, small c, small a, capital B, small c, small a, small b, small c. And these are the eggs produced. Same will be the composition of the sperms. And when we cross, we get 64 outcomes. So now students, out of the 64 outcomes developed, only one individual is a very dark individual, one individual is a very light individual, then 20 individuals are intermediate individuals. It means they have 3 dominant genes and 3 recessive genes. Here very dark it means all the dominant genes. Very light it means all the recessive genes. Then there is a dark condition where there are how many genes? There will be 5 dominant gene, 1 recessive gene, and fairly dark it means there will be 4 dominant gene and 2 recessive genes. Then there is a fairly light condition, which means there will be basically see what is very light condition. Very light condition it means all the recessive genes. Then light condition it means 5 recessive gene, 1 dominant gene, and then fairly light means 4 recessive gene and 2 dominant gene. So basically the number of the dominant gene will decide the shade color of the skin, right? Now students, again we have one chart here. So here you can see very light. So this circles indicate the pigment. So here you can see there are 6 genes which indicates that they produce no pigment. And here this particular genotype, that is a genotype of the very dark person, where all the 6 genes they are shaded with black color. It indicates what? They indicate the amount of that they produce the melanin. Now F1 generation, what was the F1 generation? Intermediate condition. See this diagram students here, that 3 genes are recessive genes which are unshaded, and 3 genes are dominant genes which are shaded black. It means they produce the what? They produce the melanin pigment. Now when we produce the gametes, so here you can see that some of them are shaded, some of them are not shaded. So here you can see these are the phenotype. One is extremely light, one is extremely dark, 20 are of intermediate condition, and then the other conditions, right? So that is basically the amount of, it depends upon the amount of melanin, and the melanin amount depends upon the number of the dominant genes. So here you can see this bar graph. This is extremely light, this is extremely dark, this is the intermediate condition where 3 dominant genes and 3 recessive genes. Here there are more recessive genes, and on this side there are more. Here you can see that there are more dominant genes, and here there are more recessive genes, right? Next slide, here we can see same thing. It will be more clear to you. So basically the intermediates, when they are crossed, same condition is explained here. Here it indicates, see the first box, it indicates that there are no basically all the genes are recessive genes, and this box indicates all the genes are dominant genes, and in between there are intermediate condition. So these are the phenotypes, right? And number of the skin alleles. Here there are no dominant genes. This condition 1 dominant gene. Here 2 dominant gene, 3, 4, 5 and 6. Here you can see that the shades of the human skin color. So phenotype and genotype and the number of the pigments or units of the pigment. So basically if a person is extremely dark, its genotype will be capital A, capital A, capital B, capital B, capital C, where all the genes will be the dominant gene. See students, here you can see that the shade keeps on changing, right? Then very dark person with only 1 recessive gene. Here you can see there is only 1 small a. So what are the units of the pigment? 5. Then dark skin, where there are how many recessive genes? Only 2 recessive genes. So see the genotype. Capital A, small a, capital B, small c, capital C, capital C. So here how many dominant alleles are there? 4. So units of the pigments are 4. Then there is an intermediate condition where 3 dominant genes and 3 recessive genes. Capital A, capital B, capital C, small c. and the unit pigments are basically 3. Now the light skin, what is the condition? Here there is a condition where there are only 2 dominant genes. See the condition, small a, capital B, small b, capital C, small c. So here you can see that there are only how many dominant genes? 2 dominant genes. Then very light condition where there is only 1 dominant gene. See the genotype, small a, small b, capital C, small c. So how many units of pigments? Only 1 unit, because only 1 dominant gene is there. And lastly, the extremely light condition where all the genes are recessive genes, that is small a, small b, small c, small c, and the unit of pigments produced is 0. So that is regarding the shades of the human color. So basically shades are dependent upon the units of the pigment, and units of pigments are dependent upon the number of the dominant genes, right? So that is regarding the human skin color, and it is an example of the polygenic inheritance. Poly, many, genic, genes. So basically many genes, they give a particular trait. What is that called as? It is called as the polygenic inheritance. Now students, the next topic, that is pleiotropy. Now in the polygenic inheritance, you have seen that a particular character is affected by more than 3 genes. Opposite to that is the pleiotropy. What is pleiotropy? Normally we know that one gene gives one character, but students, pleiotropy, it means that multiple effect of a gene. It means a single gene gives many phenotypic effect. What is it called as? Pleiotropy. A single gene gives multiple effect. What is that condition called as? It is called as the pleiotropy. So we will see the definition. Multiple effect of a gene is called pleiotropy, and such gene which have multiple phenotypic effect, what is that gene called as? Pleiotropic gene. So both the conditions are opposite. Polygenic inheritance is a single character affected by 3 or more than 3 genes, and what is pleiotropy? A single gene gives more than one character, one than more phenotypic effect. So what is that called as? Pleiotropy. So single gene exhibits multiple phenotypic expression. Now students, it is a Greek word, pleion, that is more. That is, a single gene gives more phenotypic effects. The example is phenylketonuria disease in humans. Now students, this particular disease is caused by the mutation in the gene which codes for the enzyme phenylalanine hydroxylase. So basically phenylketonuria disease is caused by the mutation in a gene which will be responsible for the formation of phenylalanine hydroxylase. So basically this enzyme called as the phenylalanine hydroxylase will who will form this enzyme? It will be decided by the gene. So gene which forms this particular enzyme, it gets mutated, right? Now what is the effect of this? So this manifests itself through a phenotypic expression which is characterized by mental retardation, reduction in the hair and skin pigmentation. So see students, basically what are the effect? Due to mutation, first of all, the enzyme is not produced. Apart from that, what phenotypic expression it shows? One is mental retardation, right? Another one is the reduction in the skin and hair pigmentation. So this particular gene, it is giving multiple effect. First effect, enzymes, as mutation have taken place, enzyme won't be secreted. Another phenotypic expression shown is mental retardation, that is first expression, reduction in the hair pigmentation second, and reduction in the skin pigmentation. Another example is the sickle cell anemia. Now students, the gene which is responsible for causing this disease is the HBS gene. Now, this gene not only causes hemolytic anemia. So, basically here what will happen? The cells of the shape of the RBCs, they become sickle shape, and as they get elongated, there will be obstruction of their passage through the blood vessels, and due to this particular cells rupture. So, what is it called as hemolytic anemia? So, now this gene not only causes the hemolytic anemia. So, this is one effect. What is the effect of this gene? Hemolytic anemia, right? But what is its other effect? It also results in the increased resistance to one type of the malaria caused by the Plasmodium falciparum. So, it not only, what are the effects produced by the gene HBS? First effect is hemolytic anemia, right? And what is the second effect? It provides the resistance to the malaria which is caused by Plasmodium falciparum. Malaria disease is caused by Plasmodium, and there are different types of the Plasmodium. One of the Plasmodium variety is the Plasmodium falciparum. So, if HBS gene is there, this gene will cause the hemolytic anemia. But apart from the causing of hemolytic anemia, what it also causes? It provides the resistance to the malaria caused by the Plasmodium falciparum. So, this gene has how many effects? Two effects. One is hemolytic anemia and another one is resistance to the malaria caused by the Plasmodium falciparum. So, a single gene is producing two effects. What is it called as? Pleiotropy. So, that is regarding the pleiotropy. What is pleiotropy? A single gene produces multiple phenotypic effect. And what is polygenic inheritance? More genes will decide a single character, or you can say a single character is decided by more than three genes. So, that is regarding the polygenic inheritance and the pleiotropy. Now students, we will move with the next topic, that is called as the sex determination. Now students, what do you mean by the term sex determination? So, it is the method by which the distinction between the male and female is established in a species. So, what is sex determination? The way through which we can distinguish between the males and females of the species, what is that called as sex determination. Now, some history regarding the sex determination. The initial clue about the sex, about the genetic or chromosomal mechanism of the sex determination can be traced back to some of the experiments carried out in insects. So, basically the genetic or the chromosomal basis of the sex determination can be traced back to some of the experiments carried out in the insects. So, experiments were taking place in the insects, and through that experiments, the genetical or you can say chromosomal basis of the sex determination was traced. In fact, the cytological observations made in number of insects led to the development of the genetic or chromosomal basis of sex determination. So, then the observations cytological, cellular, related to the cell. So, cytological observations, they were made in the insects, and this led to the development of the concept of the chromosomal basis of the sex determination. That is, through the chromosomes we can decide whether the individual is a male individual or a female individual. So, this conclusions came through the various experiments carried out in the insects. Now, history. So, first of all, contribution of Henking in sex determination. So, Henking is a scientist and his contribution in sex determination. So, Henking in 1891, he could trace the specific nuclear structure through spermatogenesis in a few insects, and it was also observed by him that 50% of the sperm received this structure after spermatogenesis, whereas 50% of the sperm did not receive it. So, Henking, he would find or he could find a particular structure in the nucleus in the insects through the process of the spermatogenesis, and he also observed that 50% of the sperms received that particular structure and 50% of the sperms did not receive that structure. Now, that structure's name was given as X body by Henking, but he could not explain its significance. What is the significance of that particular structure could not be explained by Henking. Then further investigations were made by other scientists, and they led to the conclusion that that X body which was studied or which was called by Henking was nothing but a chromosome, and that was the reason why it was given the name X chromosomes. So basically, what was the contribution of Henking in sex determination? Henking is observed in few insects that there is a particular nuclear structure, and he also observed during the spermatogenesis that 50% of the sperms they received that particular structure, nuclear structure, and 50% of the sperms didn't receive that particular structure. Basically, that structure was given name as X body by Henking, but he could not explain the significance of that X body. Later, other scientists they carried out the experiments, they investigated these structures, and they came to the conclusion that it is nothing but it is one of the chromosome, and later that chromosome was called as the X chromosome. Now, how the sex determination will be taking place. So, sex chromosomes they determine the sex of the individual in the dioecious or you can say unisexual organisms. So, basically sex chromosomes they decide the sex of an individual in the unisexual organisms. Now, all the chromosomes other than the sex chromosomes of an individual are called as autosomes. What are autosomes? The chromosomes other than the sex chromosomes of an individual are called as autosomes. Clear, students? Now, the sex chromosomes they may be similar, that is homomorphic in one sex. So, basically in one sex both the sex chromosomes they may be similar. We know that the chromosomes occur in pair. So, in one sex that sex chromosomes both the sex chromosomes are similar, that is homomorphic, and they may be dissimilar in the other, that is heteromorphic in the other. Now, those individuals which have homomorphic sex chromosomes they produce only one type of gametes, that is they are homogametic. One example we take as human beings. We know the chromosomal structure of human beings. Females have XX structure and males have XY chromosomes. So, students, here female is homomorphic. Why? Because both the sex chromosomes are similar, and males in them one sex chromosome is X chromosome and other is Y. So, what are they considered as? They are considered as heteromorphic. Now, those individuals which are homomorphic of course will produce same type of the gametes. Again, human beings females are XX. So, all the gametes produced by the females will contain only which chromosome? X chromosome. But when we consider the male which are having the constitution XY, so 50% of the gametes will receive X chromosome and 50% of the gametes receive which chromosome? Y chromosome. So, those individuals which have the homomorphic sex chromosomes, they produce only one type of the gametes, that is they are homogametic. Whereas the heteromorphic individuals, they produce two types of the gametes, that is they are considered heterogametic. See students, here a diagram is given. So this is figure A of the human beings, figure B of the Drosophila, and this figure C is of the birds. So here you can see that in the figure A and B, you can see that the female have the pair of XX chromosomes. This is a symbol for female. So here you can see that they have what? They have a pair of XX chromosome, that is they are homogametic. And what is the composition of the males? This symbol is for the males, that is X and Y, X and Y. Whereas here students, in the birds you can see that the males are homogametic whereas the females are heterogametic, or you can say the female have the dissimilar sex chromosomes and the males have the similar sex chromosomes, right? Now why the letters Z and W are given, which we will see in the later part of the chapter. Now students, we will move further. The chromosomal basis of the sex determination. How the chromosomes decides the sex of an individual. So heterogametic sex determination technique. Now what is heterogametic sex determination technique? So here in this particular method or in heterogametic sex determination, one of the sex they produces similar gamete and other sex it produces the dissimilar gametes. That is called as the heterogametic sex determination. What is heterogametic sex determination? One of the sex produces similar gametes and other sex it produces the dissimilar gamete, and the sex of an offspring is determined at the time of the fertilization only. So which type of sperm fertilizes the egg, at that time only we can decide what would be the sex of an individual. Now students, this mechanism of sex determination, there are two parts, male heterogamety and the female heterogamety. Male heterogamety is further divided into two types, XXX0 type and XXXY type, and female heterogamety is further divided into two types, ZZZ0 type and another one is ZZZW type. We will study what is both this. Here first term I have written the male heterogamety. What do you mean by male heterogamety? That is the male is heterogamete, that is male produces dissimilar gamete and female produces similar gamete, whereas female heterogamety, that is female produces dissimilar gamete and the males produces the similar gamete. Here what would be the condition? The males, they will have the dissimilar sex chromosomes and females will have the similar sex chromosome, whereas in female heterogamety, females will have dissimilar sex chromosomes and the males will have the similar sex chromosomes. Now we will study each type in detail. So first of all, the male heterogamety, that is heterogametic males. So what is it? So in this method of sex determination, the males are heterogametic, producing dissimilar gametes, while the females are homogametic, which produces the similar gamete. So basically heterogametic males is a condition where the males are heterogametic. As they are heterogametic, they produce the dissimilar gametes, and the females are homogametic, which produces the similar gametes. It is further divided into two types, XXX0 and XXXY type. So we will study each type. So first one is XXX0 type. Now students, this type of sex determination is seen in bugs, some insects such as cockroaches and grasshoppers. Now what is this type of sex determination? Here the female have the two X chromosomes. The females, they have the two X chromosome, that is they are homogametic XX, while the males have only one X chromosome. Listen very carefully. The females have two X chromosomes, so they will be of course homogametic, but the males, they have only one X chromosome, that is they are heterogametic. What is their genetic composition? X0, right? Now the presence of an unpaired X chromosomes determines the male sex. So if there is unpaired sex chromosome, what would it determine? That the individual is a male. The males with the unpaired X chromosome produce two types of the sperms. Now see students, the males are heterogametic, that is their genetic composition is X0. As their composition is X0, they will produce two types of the gametes. Half of the gametes will contain the X chromosome, whereas half of the gametes will be without X chromosome. How many types of gametes are produced by males? Two. One of the one condition is the condition where it will be having X chromosome, and other condition where the gametes won't have the X chromosome. Now the sex of the child or sex of an offspring, it depends upon what? The sperm which fertilizes the egg, because all the eggs will be homogametic, that is all the eggs produced by the females will have the X chromosome. So it is the sperm which will decide the sex of an offspring. So suppose if the sperm which carries the X chromosome fertilizes the egg, then that individual will be female. But if the sperm which is without the X chromosome fertilizes with the egg, then it develops into the male. This thing will be clear by observing this chart students. See students, this is a female. The AA indicates what? The AA indicates the autosomes, and XX indicates what? The sex chromosome. So females are having the two X chromosomes and males are having autosomes and X0. X0 indicates what students? X0 means with one X chromosome, right? Now what are all the gametes produced by the females will be AX. Now all the gametes produced by the males, in them 50% will have the composition AX and 50% will have the composition A0. As all the eggs are similar, suppose if the egg fertilizes with this particular sperm, then what would be the composition? AAXX, which indicates that the offspring is a female. But if the egg fertilizes with the sperm having A0, then the F1 generation individuals genotype will be AAX0, which indicates that the person will develop into the male. Now students, the next type is XXXY type, which is also called as the ligase type of sex determination. First of all, why XXXY type is called as the ligase type of sex determinations? Now students, this type of sex determination, it was first studied in the milkweed bug, which is called as the Lycaeus turcicus by Wilson and Stevens. Now XXXY type of determination of sex, it was first studied in the Lycaeus turcicus milkweed bug by Wilson and Stevens, and hence it is called as the Lycaeus type of sex determination. Now it is seen in human beings as well as in the Drosophila. Now students, here the females are homogametic with the chromosome, while the males are heterogametic with X and Y chromosomes. Females have XX sex chromosomes and the males have the XY sex chromosomes. Now, as the females are homogametic, they produce only one kind of the egg, and all the egg will contain what? One X chromosome. Whereas the males are heterogametic and they produce two kinds of sperms. Some sperms will have the X chromosome and some sperms will have the Y chromosomes. Now, sex of the embryo will depend upon what? Upon the fertilizing sperm. Which sperm fertilize? See, eggs, all the eggs are same. All the eggs contain what? X chromosome. But it is the sperm where some of the sperms carry X chromosomes and some of the sperms carry Y chromosomes. So, which type of sperm fertilizes the egg will decide the sex of an individual. An egg fertilized by X-bearing sperm, that is, if an egg is fertilized by a sperm which carries X chromosome, it will develop into a female. Why? Because two X chromosomes. But if an egg is fertilized by a sperm which contains a Y chromosome, then it will be producing a male, because there is one X chromosome and one Y chromosome. See the chart, student. The parents are, this is a female parent, that is AAXX. AA indicates the autosomes, right? XX indicates the sex chromosomes. The females are homogametic and this is the male with the composition AAXY. Now, all the gametes produced by the females have the composition AX, whereas the gametes produced by the males, so all the 50% of the sperms will have the composition AX and 50% of the sperms will have the composition AY. Now, which sperm fertilizes the egg will decide the sex of an offspring. Suppose an egg is fertilized by a sperm having the AX chromosomes, then it will be AAXX, which means that the individual is a female. But if the egg fertilizes with the sperm having the AY composition, then the genotype of the individual will be AAXY, and as it is XY, it will develop into the female. So, this is the condition of the heterogametic males. That is, males produces the dissimilar gametes and the females are homogametic, that is, they will produce the similar gametes. Now, students, opposite to that is the heterogametic females. So, what do you mean by heterogametic females? It means the female will produce the dissimilar gametes and the males will produce the similar gametes. So, students, in this method of sex determination, the homogametic male possesses two X chromosomes as in certain insects and certain vertebrates like fishes, reptiles and birds, producing single type of gamete, while the female produces the dissimilar gametes. So, what is heterogametic female condition? Here the males are homogametic, that is, they produce the similar gametes and they will have the two same sex chromosomes, whereas the females are heterogametic, it means they produce the dissimilar gametes. So, here the female consists of single X chromosome or one X chromosome and one Y chromosome. So, what would be the composition of the females in these conditions? So, either the female will have only one X chromosome or they may have one X chromosome and one Y chromosomes. So, here females are heterogametic and they produce how many types of egg? Two types of egg. Whereas in the heterogametic males, the eggs were produced of only one type and the sperms were produced of two different kinds. Here in the heterogametic males, the sperms are of one kind but the eggs produced are of two different types. Now students, here to avoid the confusion with XXX0 and XXY types of sex determination, in the heterogametic males we have studied XXX0, XXXY type, right? So here to avoid this confusion we will be using the word Z and W, right? So here the word alphabet Z is used instead of X and W is used instead of Y. So to avoid the confusion with XXX0 and XXXY types of sex determination, the alphabet Z and W are used here instead of X and Y respectively. So here heterogametic female, they are of two types, Z0 ZZ type and ZW ZZ type. So we will study each one in detail. So the Z0 ZZ type. So here it is seen in certain moths, butterflies and domestic chickens. So here the female, it possesses only single Z chromosome. Okay, the female will have only single Z chromosome and it is heterogametic. So its composition is Z0. So how many types of egg will she produce? Two types. Some which contains the Z chromosomes and some without the Z chromosomes. Whereas the male possesses two Z chromosomes and is homogametic, that is ZZ. So as it is homogametic, it will produce all the sperms which will contain the Z chromosomes. So chart we will study. So this is a female. A indicates the autosomes, right? All the chromosomes apart from the sex chromosomes, they are called as autosomes. So AAZ0, Z0 it means the female. So it produces how many types of gametes? Two types of the gametes. Some eggs will contain the Z chromosome and some eggs won't contain the Z chromosome. Whereas these are the males with the two Z chromosomes. So all the sperms produced by the males will contain the Z chromosomes and all the gametes will be same. Now students, which type of the egg gets mixed with the sperm will decide the sex of an offspring. So suppose a sperm is fertilizing a egg of AZ. So basically suppose AZ egg, it fuses with the sperm, then the offspring will be what? AAZZ, that is it will develop into a male. But if a sperm fuses with the egg which is without the Z chromosome, then the genotype will be AAZ0, right? And it will develop into a female. The next type is ZWZZ type. So it is basically seen in the insects like gypsy moth and some vertebrates like fishes, reptiles and birds. So here the female has one Z chromosome and one W chromosome. So its composition is ZW. As it is heteromorphic, it produces two types of the egg. Some eggs will carry Z chromosomes and some eggs will carry the W chromosome. Whereas the males contains the two Z chromosome, that is it is homogametic, ZZ. So it produces a single type of sperm and all the sperms will contain what? The Z chromosome. Now chart we will see. So this is a female with a ZW, this is a male with ZZ. The female produces two types of the egg. 50% eggs contain Z chromosome, 50% of the egg contain W chromosome. Whereas all the sperms produced by the males will contain the Z chromosomes. Now suppose a Z chromosome containing egg fuses with the sperm, then it will develop into, basically there will be 2 Z chromosome and it will develop into a what? It will develop into a male. But if a egg containing W chromosome fertilizes fuses with the sperm, then it will have the composition ZW and it will develop into a female. So that is the heterogametic females, where the males produces the similar sex chromosomes and the female produces the basically the males produces the similar gametes and the female produces the dissimilar gametes. Now we will study the sex determination in the human beings. Now, genes determining the sex in human beings are located on the 2 sex chromosomes called as allosomes. So, students, sex chromosomes are called as allosomes and all the chromosomes except the sex chromosomes are called as the autosomes. Now, in mammals, the sex determination is associated with the chromosomal difference between the two sexes. We have seen that the XX females and XY males. 23 pairs of the humans include 22 pairs of the autosomes, that is 44A. A indicates autosomes, and 1 pair of sex chromosome which may be XX or it may be XY. Females in the humans are homogametic, which produces only one type of the egg, each containing one X chromosome, while males are heterogametic, producing 2 types of the sperm with X and Y chromosomes. We will see the table. So basically this is a human male which is heterogametic, this is a human female homogametic. So males, they have 44 autosomes plus XY sex chromosomes, and the females have 44 autosomes and XX sex chromosomes. Now the sperms produced by the males, they will contain 50% of the sperms will have 22 autosomes plus X chromosome and 50% will have 22 autosomes plus Y chromosome, whereas all the ovum or eggs produced by the female will have the composition of 22 autosomes plus X. Now, what would be the offsprings or the progeny? If X containing sperm fuses with the egg, it develops into a female. If Y containing sperm, it fuses with the gamete, it will produce a male. So, all the possibilities, what is the possibility? 50% chances are for the development of the females and 50% for the males. If this fuses with this, suppose 22A plus X fuses with 22A plus X, it will develop 44 autosomes plus XX, that is a female. 22A plus X, if it fuses with the 22A plus basically X, again it will produce what? 44A plus XX. But if 22A plus Y fuses with 22A plus X, it produces 44A plus XY, that is a male, and here also it will produce a male. So, there are 50% chances that males will be developed and 50% chances that females will be developed. But see students, all the gametes produced by the females, they are similar. It is the sperms which are having 2 different types, one with the X chromosome and one with the Y chromosome. So, which type of sperm fuses with the egg will determine the sex of an offspring. Now, we have already mentioned that sex determining mechanism in case of the humans is XY type, right? So, out of 23 pairs of the chromosome present, 22 pairs are exactly same in both males and females. They are called as autosomes. A pair of X chromosomes are present in female, whereas the presence of the X and Y chromosomes are determinant of the male characteristic. Now, during spermatogenesis among the male, two types of the gametes are produced. 50% of the total sperms produced carry X chromosome and the 50% has the Y chromosomes besides the autosomes. So, but the females, however, they produce only one type of ovum with the X chromosome. So, there is an equal probability of fertilization of the ovum with a sperm carrying either X or Y chromosomes. In case the ovum fertilizes with the sperm carrying X chromosome, the zygote develops into the female, that is XX, and the fertilization of the ovum with a Y chromosome carrying sperm, it results into the male offspring, that is, it will have the composition X and Y, right. So, it is evident that it is the genetic makeup of the sperm that determines the sex of the child. So, who decides the sex of the child? Which type of sperm fertilizes the egg? If a sperm carrying X chromosome fertilizes the egg, then it will develop into a female child. And if a sperm carrying Y chromosome fertilizes the egg, it will develop into a male child. So, it is also evident that in each pregnancy there is always 50% chance or probability either of a male or a female child. But it is unfortunate in our society that always women are blamed for producing a female children, and they have been ostracized, that is, they have been excluded from the society and ill treated because of this false notion. See students, through this we came to conclusion that it is the sperm which fertilizes the egg decides the sex of an individual, right. Suppose if a X carrying sperm fertilizes the egg, it will develop into a female, and if a Y carrying sperm fertilizes the egg, it will develop into the male. So, basically it is the genetic makeup of the sperm which decides the sex of a child. But it is very unfortunate in our region that normally women are blamed for the female children, but it is not the women which decides the sex, it is the sperms which decides the sex, and due to this false notion the women have been ostracized. It means they have been excluded from the society and they are also ill treated. Now students, the sex determination in the honeybee. Now in the hymenopteran insects such as honeybee, ants and wasps, a mechanism of sex determination is called as haplodiploidy mechanism of the sex determination. What is it called as? Haplodiploidy mechanism of the sex. Now what is that? We will see. So here the sex of the offspring is determined by the number of the sets of the chromosomes it receives. So how many sets of chromosomes are present will decide the sex of the individual. Now students, listen very carefully. An offspring which is formed by the fusion of the sperm and egg, or basically you can say a fertilized egg, they develop into the female. So if an offspring is a result of fertilization, that is, if offspring is formed by the fusion of sperm and egg, it will always develop into the females. Now that female may be a queen or it may be a worker, and the unfertilized egg, they develop into the males or called as the drones by the process of parthenogenesis. So if fertilization occurs, always it will develop into the female, and if the unfertilized egg is developed into the individual, then it always develops into the males. It means we can say that males have half the number of chromosomes. Why? Because they are developed without fertilization. So they are always haploid, and the females have double the number because they are formed by the fusion of the egg and sperm. So they are always diploid, and hence the name is haplodiploidy for this type of the sex determination. Why it is called as the haplodiploidy sex mechanism? Because males have half the number of chromosomes, that is they are haploid. Females have double the number of chromosomes, that is they are diploid. Now students, the females, we are talking about honeybee. So the females, they are diploid, they have the 32 chromosomes, and males are haploid, that is they will be having only 16 chromosomes. This type of sex determination is called haplodiploid sex determination and has the special characteristic features, that is sperms, they produce the males, they produce the sperms by mitosis. Why? As the males are haploid, they always produce the sperms by the process of mitosis. They do not have the father, right? Of course, the males won't have the father. As they don't have father, they cannot have sons. Yes, but they do have a grandfather and can have the grandsons. See students, the diagram, the female with the 32 chromosomes, male with the 16 chromosomes. The female by the meiosis produces the eggs with the 16 chromosomes, and the male by mitosis produces the sperms by containing the 16 chromosome. Now suppose if this particular, see the condition, this is a female. Now if the fusion occurs between these two, then it will develop into a what? It will develop into a female. But if this unfertilized egg directly develops, then it will develop into a male, which will have the 16 chromosomes. See, the next slide will be next clear. Female, which is a queen, which have a diploid chromosomes, and the males, they have the haploid chromosomes. Female by the meiosis produces the egg, each egg containing 16 chromosomes. Males by the mitosis produces the sperm, each sperm contain 16 chromosome. Now suppose this is the egg which undergoes parthenogenesis, that is no fertilization, and it develops into the individual. Then it will develop a haploid male. But if the fusion occurs between the egg and the sperm, that is fertilization occur, then it will be a female with the diploid chromosomes. Now this female either may be a queen or it may be also the worker. So that is the process of the sex determination in the honeybee. So here we come to the end of our topic sex determination. Next topics we will be clearing in, or we will be studying in the next lectures. So today we have studied polygenic inheritance, pleiotropy, as well as we have studied the sex determination methods. I end my lecture here. The next topics we will be studying in our next videos. Thank you students.